When it comes to the eye, most people are familiar with the outer segments such as the lens, pupil, iris, and cornea. However, what goes on in the back of the eye, specifically in the retina, is a much more complicated process. So let's look at what exactly goes on. When light hits the retina, the first set of cells that respond are called the photoreceptors. They send a signal to the bipolar cells, which then send a signal to the ganglion cells. From here, the signal is sent up to the brain for processing. For my research, I'm focusing on the synapse or the connection between the photoreceptors and the bipolar cells, so let's zoom in there. There are two types of photoreceptors, cones and rods, and there are two types of bipolar cells, on and off type. Typically, cones synapse with both the on and off type bipolar cells, whereas rods only synapse with the on type. The connection in the on type pathway is mediated by a protein called mGluR6. If we zoom into the synapse, we can see that mGluR6 is a G protein coupled receptor. It regulates the activity of an ion channel known as TRPM1, which allows for the passage of sodium and calcium ions. In the dark, glutamate is released from the photoreceptors and it binds to mGluR6. This causes a conformational change in mGluR6 which activates the G protein. The G protein then has an inhibitory effect on TRPM1, causing it to close and thus preventing the passage of ions into the bipolar cell. In contrast, during the day, there is no glutamate release, so mGluR6 and the G protein remain inactive. This allows TRPM1 to open and ions pass through into the bipolar cell. So now the question to ask is, what would happen if mGluR6 and this pathway did not work? As it turns out, a condition known as CSNB, which stands for congenital stationary night blindness, has been linked to defects in mGluR6. In particular, 16 different missense mutations have been found in patients with CSNB. The mutations are split across the protein's three distinct domains: the ligand-binding domain, where glutamate binds to the protein, the cysteine-rich domain, and the transmembrane domain, which is responsible for anchoring the protein to the plasma membrane and binding the G protein. So the question I asked for my research is, what are the molecular effects of CSNB-associated mutations in mGluR6? To start, I looked at mGluR6 localization HEK cells using immunofluorescence microscopy. I found that firstly, mutations in the ligand-binding domain cause varying levels of surface or plasma membrane expression. Compared to the wild-type or normal condition, some mutants have the same or slightly higher level of expression, whereas others have reduced expression. The sole CRD, cysteine-rich domain mutant, has found to have reduced levels of mGluR6 expression compared to wild-type, and as for the transmembrane domain, all of the mutants had lower levels of surface expression compared to the wild-type, yet the degree of the reduction varied. With the mGluR6 mutations having varied effects on localization, there is room for further experimentation, such as testing for synaptic localization in mice and for protein function. Perhaps one day in the future, these findings can serve as a basis for clinical studies aimed at treating night blindness, turning impaired vision like this into this.